On the one hand, we have this proposed method to reduce our anthropogenic impact on the climate. On the other hand, from a more local perspective, we have that mitigation technique directly affecting a very small area where we all live and care about.
Let's face it, there's South Louisiana has a long history of having bad environmental stuff done to it. I mean, a long, long history. And of governments and companies saying, oh, it'll be fine, and guess what? It's not fine. And the people and the area, the environment ends up paying for it. And when in South Louisiana, when the environment's impacted, people are impacted. And we are very, very closely related to the environmental, health of this area here.
We at Southeastern are very happy to be involved as an independent monitor of what's going to happen over the course of these next few years as companies try to get the permits to do the carbon sequestration. And so what Southeastern, our biology department, and Turtle Cove are going to be involved in is looking at the biological impacts of whatever is happening with the work, both before and if they get the permits later on during the carbon sequestration process of putting liquefied CO2 down 1.5 miles under the surface.
Right now we are contracted for a 3-year monitoring project where we monitor the aquatic environment and the organisms in it. We monitor the wetlands in the area around the lake. We will monitor the chemical composition in the water column, and we will conduct some education and outreach. Sort of the 4 components of this project. We wanted to be involved to serve as the independent monitors for this project. Obviously, this lake is in our backyard. We have the expertise to work there, and we felt that it was good for us to be involved as long as we were allowed to remain independent. We have the ability to control the data. We own the data. We're going to make the data publicly available on a website so that anybody in the public can log in and look at, let's say, the dissolved oxygen concentration in the lake, the temperature of the lake, the pH, any of those parameters, as well as the biological data that we'll be collecting over the next 3 years. So it's really important to remain independent. I will be doing something called environmental DNA monitoring, which is a technique where we go out and we sample water. As organisms are in the aquatic environment, they shed DNA, scales, mucus, feces, urine, that puts DNA out into the wild. We can capture that DNA in a water sample through filtration. We take those samples, those filters that have the DNA on them, and we use fish-specific primers or probes to amplify the fish DNA, simply based on the presence of the DNA they've left in the water column, their signatures. We're also going to be setting crab traps to monitor crab populations. We'll be trawling for shrimp populations, and we'll be using dredge grabs to capture Rangia clams that live in the sediment. You go out to Lake Maurepas on any given day and there are hundreds of crab traps set out there. But you try and get the historic data from the state agencies or from the feds, it doesn't exist. So we're going to be able to provide baseline data on population dynamics of blue crabs. How big are they? Male-female ratio. What's their growth rate? Things of that nature. We simply don't know about Lake Maurepas. In addition, Lake Maurepas is a shrimp sanctuary. So nobody can shrimp there. What does that mean? We don't know anything about the shrimp populations in Lake Maurepas. So we're going to be able to gather that information on abundances of shrimp in Lake Maurepas, something we currently don't have. So these are examples of new data that we're going to be able to gather and analyze, make available to the public, and then hopefully publish in a scientific journal to make it available in literature forever. That's our goal, is to gather that baseline data that we simply don't have.
So we're studying crabs, frogs, catfish, and alligators. And the reason we're studying those 4 animals is because predominantly we're interested in how biochemical contaminants may affect all biota, living organisms, in particular animals in Lake Maurepas.
sampling where I'm at, and I'll take data. Just hold it down.
So 3 of the main response variables that we're going to be utilizing or collecting data on across all 4 of our species are chronic stress, sex steroid quantification, and liver and reproductive organ histology. We don't know a lot about, for instance, chronic stress levels of these organisms in Lake Maurepas. Are they already being stressed out physiologically by something? We don't really know about their reproductive energy allocation. How many offspring are they having? Are they reproductively very healthy? We don't know what pathologies currently exist in the lake in these organisms. So I think that getting a really good handle on where we're at in the lake from an organismal health perspective is the only rational first step to be able to diagnose potential alterations of that health later on.
I think that the reciprocal
As
algorithm.
a scientist, I aim to evaluate these data to be certain that negative impacts aren't occurring, and when they are, bring them to light as soon as possible so that we can mitigate these problems.
The vast majority of Maurepas is swamp, not marsh, and the 13% of it that's healthy is healthy for a very specific reason, and that is because the water is moving. So as long as the water sort of pushes through neighborhoods and out into the swamp, you've got really nice looking trees that are truly sustainable. But the vast majority of the swamp is in fact dying. And in fact, in an 11-year period when we studied the swamp from 2000 to 2011, over 33% of the swamp we were studying, the trees that were tagged with our tags, died. So one of the things we're doing every year is planting 2,000 seedlings. So first time in 60 years, we're seeing seedlings jump out of the ground everywhere. We have 6-year-old saplings now that are 12 to 15 feet tall. It's just so wonderful to actually see that ecosystems in coastal Louisiana can be restored. So we're measuring the ecosystem response to whatever its environmental conditions are. So is it growing or not growing? And if it's growing, how much is it growing? And does it grow mostly from leaves, or does it grow mostly from wood? Or is it 50-50 leaves and wood? All of this stuff is important with regard to deciding whether the restoration worked or not.
We will closely monitor the water quality by sampling weekly from different depths of the lake. The lake has different depths, depends to which location you look into, but we will collect waters from the ground, from the surface, and in between. Having this data will help us to figure out what is going on in the lake. Is toxic chemicals are increasing? Are they decreasing? Is going to be any changes to their concentrations? Are we going to have new compounds or not? We'll have buoys in 4 locations of the lake, and that will help us to monitor carbon dioxide as well. By working on this project, I am hoping for the first time we will have a deeper knowledge about the lake, about the chemical composition of the lake, and that will help us to come up with better strategies and help to in terms of the well-being of the lake and the environment in general.
So our involvement in this project is really essential so that we can determine if there are impacts of any of their activities. Because in all honesty, we simply don't know whether there'll be any impacts at all. Our involvement is imperative to determine if that's the case or not.
So our job is to protect our home in that sense, right? Our job is to elucidate potential negative impacts that this project has that may go unseen for a long time.
We're going to be putting all of our findings out for everybody to look at. We're not going to hide anything, and so whatever happens, we will know it happened, and the public will know it happened.
We want to know if there's things going on as a result of their activities. We want to be the stewards of the lake.